Hello, my name is Shitij Kapur. I'm the Dean of the Faculty of Medicine, Dentistry and Health Sciences and the Assistant Vice-Chancellor of Health here at the University of Melbourne. As the coronavirus takes its toll internationally, my thoughts first go out to the millions of Chinese citizens who find themselves locked in their own cities and homes in an effort to stop the spread of this virus. One can only imagine the disruption and the anxiety and the fear in their lives. My special thoughts go out to the so many of our students who are now caught on the wrong side of the travel ban. So many of you who were enrolled in our courses went home to China to celebrate the Lunar New Year, but find that your plans to come back have to be put on hold. But also to the many of you in your first years who must have been excited to come to the University of Melbourne, but now find yourself in a limbo. We understand that this may be an anxious and concerning time for many of you. Please check the website on the screen, which will have our latest updates. You can also access counseling support online to help you through this difficult time along the link that is provided. That said, I remain confident that we will overcome these challenges with science and solidarity, and that is what brings me here to the Peter Doherty Institute at the University of Melbourne to hear one of the spectacular success stories in the international campaign against this new virus. So first, let me speak to Dr. Mike Catton and Dr. Julian Druce. Welcome, Mike and Julian.
Hello.
Could you introduce yourself to the audience and tell them what you do?
Certainly. I'm Mike Catton. I'm Deputy Director of the Doherty Institute, and within that I'm Director of the Virology Public Health Lab, which is VIDRL for short, but its long name is the Victorian Infectious Diseases Reference Laboratory. I'm a clinical virologist, so I work in a lab, but I am working on patient specimens and in public health, so that's my specialty.
Wonderful.
I'm a pathologist by trade.
Thank you. And Julian?
So I work in a laboratory within the Victorian Infectious Diseases Labs called the Virus Identification Lab, and I'm a diagnostician, so I design and develop diagnostic tests for various pathogens, but mostly viruses.
Well, it's a wonderful collaboration between a clinician and a scientist.
Yep.
And of course, everyone in the world now knows that your teams were the first teams outside of China to grow this novel coronavirus.
Yes, to grow and share.
To grow and share,
grow
as
and
you
share.
say. Well, you know, to most of us, we don't think about growing viruses, we're just afraid of catching them. So how do you grow viruses safely?
Yes, so the safety angle is linked to risk group, and so there's different levels of risk for viruses. So some are more contagious than others, but in essence, all of the culturing is done by classical virology techniques, which is on to cell cultures, which are basically a single cell layer of different types of cell lines, and these have different receptors, so there's many different cell lines available, and this was the traditional methods 30 years ago, 20, 30 years ago, to diagnose patients with a virus, that you'd put the patient sample onto these cells and actually look for the virus replicating.
So you put the sample onto cells and then you see if you can see viruses growing?
We don't see the virus per se, we just see the effect that the virus has on those cells.
And from that you know that viruses have started growing in this culture.
Yeah.
And even I'm old enough to have grown up with that as diagnostic virology technology.
That used to be.
It used to be the way that viruses were diagnosed, often 3 weeks after the patient had got
And it
better.
used to take a long
Very
time
slow
to do that.
Yeah.
Now, of course, as soon as this coronavirus came out and the rest of the world started to get knowledge of it, I would assume there were dozens of labs trying to do what you were able to achieve. How do you think you got there first?
Look, I think chance favours the prepared mind, so to speak, in that our role as a leading public health laboratory is to be prepared for the unexpected, not just to react to things as they happen. So I think we as a group, Julian as a diagnostic scientist, we're of the view that SARS and MERS would not be the only coronaviruses that we would see emerge probably from animals into the human population.
So you've just used here two words, SARS and MERS. You might want to tell our audience, most would know SARS, but MERS?
Yeah, MERS is a relative of SARS, another coronavirus, and it spread in the Middle East from camels, and it sporadically jumped from camels into humans in the Middle Eastern part of the world, causing severe disease, even more severe than SARS.
So you were anticipating that something like this might happen. You were prepared for it.
That's right. So Julian had designed broadly reactive nucleic acid or molecular tests that would detect all human and animal coronaviruses. Because his team had used international genetic databases to look at the sequences of all of those viruses and to predict reagents that would detect all of them. So that was a key piece of work that Julian did. And then as well as that, we had rapid gene sequencing capability to help us confirm results in the middle of the night, and the famous culture flasks of susceptible cells that are there 24/7.
So they were ready. So you had all the reagents ready. You were scientifically prepared to find this, but still there has to be a moment when it actually happens. So walk us through the challenges and the excitement, because I would presume that to grow the virus you had to get a patient sample in the first place.
Yeah, so obviously we have to have a positive sample.
Yes.
So we firstly identified that on the Friday night, and then we confirmed it through into the wee hours of the morning on Saturday.
Hang on, so Friday night you got a sample, you knew that this person has the disease, but as you said you couldn't see the virus.
That's right. So we detected it by these molecular fast mechanisms of detection. So we pretty confident. But
so Julian, you should tell the story, because I think it's a story worth telling, because it's just that typical scenario of the Friday night before a long weekend. It was
right. It was the Australia Day.
The Australia Day weekend.
Right.
So as I've said in a couple of situations in public, Julian and I and my registrar were just sitting down after I'd been in a lengthy teleconference. It was sort of last thing Friday afternoon. We were facing a 3-day weekend. And we were just sitting down to talk about the staffing of the lab and preparations for that long period of sort of skeleton staffing, but knowing that there would be a test demand on us. And we'd really scarcely sat down, then a couple of Julian's team were hovering in the door saying, "We think we've got a positive." So, and then we thought, there goes the weekend. And it really didn't disappoint.
Well, so it was the weekend that hopefully changed
and at that point the test is unvalidated. This is an unvalidated diagnostic test, so it's all working in principle, and we have that expectation, but we have to prove, as scientists we always like to prove. So we have the secondary tests, which were these ones that Mike just said that we developed 10 years ago for this capacity to discover something new. So that was the secondary test, with the expectation that the first result was genuine, and with that result we put that sample, that patient sample, or 500 microliters, half a mil of that sample, onto these cells, a flask of cells that are growing on a plastic tray, and
waited.
So
and I should interject, it isn't an abstract exercise either, because there's a patient, there's a clinical team, there's the health department, everybody that needs to swing into the action to manage a clinical and public health scenario. So as we're getting that first unvalidated result, then communications are having to happen with people that need to know to care for the patient, to manage possibly the public health consequences, while Julian's team rolls on.
Right, so that's the sort of collaboration between science and clinical.
That's right.
You know, you're breaking new scientific ground, but on the other hand you have the obligation to get back to the patient to help their care. But tell me about, I almost have the sense it's like baking the cake. You're watching the oven, it's not arising. How long does it take for you to know that there is virus in the sample that you've been able to grow?
So we're lucky in this day and age that we have these additional molecular tools. So the same test that we diagnose the patient with gives us a signal of the burden of virus in the patient sample and both in the culture. So at 40 hours into the incubation, so this was Sunday morning of the long weekend, I went in and I sampled from that culture, and it was a little bit more positive than the patient sample, but not a lot. So it's effectively 4 times more than the signal from the primary sample from the patient.
So you saw that there was a signal?
So at that time, the look of the cells was tantalizing.
Yes.
So I placed the cells onto a small microscope that had a camera, and it takes a photo every 15 seconds and assembles those photos into a video file. And so that's the file that's been released, and it
that's
shows
a file
the
that's
progression.
gone viral all around the world.
Correct.
And everyone's looking at that, but they're trying to find the virus. They think, where is the virus in all of this? So how should we look at that data?
So as that video progresses, at the beginning or commencement of the video, the cells look healthy and normal.
Right.
And that is looking at the cell sheet. And so you can see individual cells there, and they kind of wobble a little bit. It's surprising, but they do move around. And as the video progresses, there's black dots that appear. And this is actually cells that are infected with the virus dying and kind of lifting off the cell surface, the sheet of cells, and coming up into the medium.
So one isn't actually seeing the virus, one is seeing the virus killing the cell.
Correct.
And one is seeing the death of the cell.
That's right. The virus is way too small to see by normal light microscopy. You have to do electron microscopy to see the virus.
So then how do you then confirm that this was the coronavirus that was killing the cells?
Yes, so firstly we know that the virus is there because of the cytopathic effect, or the cellular changes that are occurring. And then to prove that it's the virus, we sample from the fluid from the cells, and it's very, very strong by that stage. At the end of that video we sampled, and it was then 1,000 times stronger than the original patient material. So we had amplified the virus at least 1,000 times.
Now, and what time was it when you were then confirmed and you knew that you had done it?
So that was actually when the ABC was visiting.
Right.
And so it was just fortuitous that they'd already been engaged to come to the lab prior to even the first detection. They were just coming to do a news story about diagnostic capacity.
In general,
yes,
because of this, and it was there. What a lucky coincidence that is.
That's right.
And now what happens in a lab when something like this happens for the first time? Is there a ritual? Do you like pop a champagne? Perhaps you can't do that in your PC lab, so maybe come outside and pop a champagne.
No.
We just get on with it. And,
sadly,
we're just happy that we've got it, and we knew that no other lab, because we'd been on these World Health Organization teleconferences pretty much every second night from the commencement of real concern. So this is from mid-January, or about the 11th or 12th of January. And WHO were very, very interested in an isolate, because at that point I think things were going downhill rapidly in China, and so there was no capacity for them to share.
Right. Now WHO, of course, is the World Health Organization, and so you said something very important. You said you were not only the first group to grow, but grow and share.
Yes.
So tell me about the sharing part. So now you've identified the virus, you've figured out how to grow it.
Mm-hmm.
Do you now make it industrial? Do you now start making a lot of it?
I rang Mike on Monday morning and I said, look, I'm involved with, he knows I'm involved with a lot of the meetings with WHO, and they're pressuring us to share or get the virus. And we agreed that it was very important from a public health perspective to share that virus and be transparent and open, because it leads to so many other things.
It's wonderful that you've done that, but for the rest of us who are hearing, it's a little bit worried sharing a virus. So tell us how it's done safely, and how does the virus safely go around the world?
Of course.
Unlike the unsafe transmission.
To backtrack a little bit, Julian and I have lived through earlier health emergencies. We've had the experience with SARS, a close relative of this virus, and how hard it was, even then, and things have become more difficult, to share reagents and particularly viruses between expert labs, just because of paperwork and regulation that is important in some ways, but hampering the public health effort in an emergency. So I think it was really important that not only was the virus grown, but it was shared early through the advice of the World Health Organization, so an expert international authority who could direct us to who they thought the priority laboratories were. So yes, through international culture collections, we can make the virus broadly available to researchers and everybody that needs it, that are well credentialed. But in the first instance, this is about supercharging the response. So there are certainly really expert leading coronavirus reference labs internationally.
Right.
We know who some of those are. They're regularly in contact through this emergency. WHO could guide us to who are the top few that should get it.
And you work
Correct.
with them.
And then to answer your question, there are really detailed international standards for how things are safely moved around.
I'm sure they are.
Regulation, and physical.
We feel comfortable that we're in the hands of experts who know how to do this
Good.
very, very well. But let me understand, though, that while this was happening, of course, in Australia, there was concern about new cases, but you also serve as a reference lab. So there
Yes.
were also other Australian labs
Yes.
that were sending you samples so that you could confirm and help them develop
Indeed,
their assays.
yeah.
So tell us about that.
Well, really only this week, now 3 weeks after that period, only now are we relinquishing responsibility for most of the other Australian states and New Zealand to be providing either direct testing or confirmation. Because of the time it's taken to get.
But but over the last 2 weeks, they were working with your lab
us,
to
yeah.
build up their capacity to test across Australia and New Zealand.
For some things in Australia, and there are official national reference labs, and we are that for a number of other viruses, and Ebola is a particular one that might resonate with people. We have the high containment suit lab for Ebola, and we are the designated national reference lab for that. For coronavirus, there's no such thing as a national reference laboratory.
Well, they couldn't have been before this, so now you are it.
But it just happens that I would say we're the first amongst equals. Really, in terms of our capacity and stature and the leading role we often play, which is linked to the suit lab in many ways, because we've got the capacity to pretty much do anything with anything, and that leads to a kind of first amongst equals role. So with this, it was equally true that we were perhaps the best prepared out of the starting blocks with our tests and our capacity and our expertise. So not by anyone saying, now you're in the lead, just by assuming the role and kind of taking the leadership. We were then providing the diagnostic capacity directly for many states that didn't have it, and for states like New South Wales who had some testing capacity but maybe weren't as confident with the results as we were. They wished to refer to us for theirs to be confirmed.
So, and testing is one thing. We were also very lucky that the first positive sample came to us as well.
Yes, that's true too.
So that's a big element of the equation.
So all right, so we know how to test, we even know how to grow the virus. Tell us how having grown the virus helps us find treatments and vaccines.
Well, look, the Doherty Institute was conceived, the idea and the vision really was about this sort of scenario, which is about somewhere in the high quality public health that's being done either by us or our sister lab, MDU, operated by Melbourne University, that there are important public health findings made, but in our past experience is limited capacity to really exploit and translate and take the next step. So instead this time we have around us all those expert minds, all of those facilities, all of that capacity and research, epidemiologists, clinicians, all of the skill sets really to take anything from the bedside through to the bench. A multidisciplinary team could be assembled here to tackle pretty much anything.
So what would be your guess? You know, often I find in many of these epidemics, we find the vaccine or the treatment after the epidemic is gone. How long might it take this time? Now, it's a hard question, but you know, your guess will be better than mine, so better yours.
I'm not a vaccinologist. We have other people more expert than me in that. I know there are a number of labs internationally that have platforms that would facilitate rapid vaccine production or development and production responses. So I think we will certainly see a coronavirus vaccine more speedily than we might have seen candidate vaccines for other agents in the past. It'll still take quite a long time, many months. And perhaps It's certainly true that many outbreaks are resolved by public health practice and action, and the vaccine comes along as a second level issue.
And of course one needs that as
Correct.
Life goes on.
So whether this outbreak lasts long enough for the vaccine to come and make a difference.
First of all, it's been a privilege, through your eyes to get into the excitement of science, but also to hear how it actually is impacting upon clinical care through your laboratory work right now. So thank you for letting us into that world.
Thank you.
Thank you for all that you did. Perhaps you might also thank your families on our behalf for the long weekends you missed. But we are really delighted to be associated with the fine work that you've done.
Thanks.
I guess you'll now run and do some more.
Of course. And look, this activity is a team event and not a solo activity, so there is a team of people that have worked through the night and are working long hours and through the weekend. So I'd like to really thank all of them as well.
Well, thank you very much.
Cheers.
Well,
Thank you very much.
To discuss the implications of coronavirus for individuals and for public health, let me turn to Professor Kanta Subbaroa and Professor Sharon Lewin. Welcome, Kanta, and welcome, Sharon. Could you just introduce yourselves and tell the listeners a little bit more about what you do here?
Sure. So my name's Sharon Lewin. I'm the director of the Doherty, which is a joint venture of University of Melbourne and Royal Melbourne Hospital.
But Kanta, about yourself?
Sure. I'm Kanta Subbaroa. I'm the director of the WHO Collaborating Center for Reference and Research on Influenza that is based here as part of VIDRL, as part of the Doherty Institute. So I'm an influenza virologist. I trained as a physician in pediatric infectious diseases and also spent several years working on SARS coronavirus and a few years on MERS.
So let's start there, because people heard about the SARS and then there was a MERS, and not everyone heard about MERS. And now there's this new virus. Mm-hmm. Are they related in some way?
So they all belong to the same family of coronaviruses. So coronaviruses come in families, and the classification depends on a number of things, including the nature of the nucleic acid, which is their genome, and other characteristics in the genome organization, the proteins, and so on. So all three of these viruses belong to the family of coronaviruses. And many, many animal species have their own coronaviruses. So until 2003, we did not believe that animal coronaviruses would cross the species barrier because they tended to stay within their species. So 2003, with the emergence of SARS, was the first time that an animal coronavirus infected humans. It was also the first time we had severe infection associated with the coronavirus in humans, because all of the coronaviruses that infect humans have generally caused very mild illness. They cause a cough and a cold. So the 2003 outbreak was SARS, and that was controlled with public health measures. No vaccines were available and no drugs were available. And then 10 years later, MERS emerged, again from an animal host.
So these coronaviruses are all related. From what we know today, how is this new coronavirus, which seems to have started in Wuhan, different from the previous one?
So the coronaviruses, as I said, they are a very large family of viruses, and they've been classified into alpha coronaviruses, beta coronaviruses, gamma, and delta. Many of the human coronaviruses are alpha coronaviruses. There are a few beta coronaviruses. SARS is a beta coronavirus, as is this novel coronavirus. So the novel coronavirus is about 70%, shares about 70% identity with SARS. So it is not SARS.
So it's not SARS?
It's not SARS. It belongs to the same family and appears to use, or at least bind, to what we know is the receptor for SARS.
Is it more serious than SARS?
That's a complicated question. The case fatality, which is how we usually think about whether something is serious.
So case fatality is number of people who get the virus
who
and die.
Yes. So in a case of if somebody is infected and they get severely ill and die, the case fatality refers to the number of people or the proportion who die having been identified as a case. So the case fatality rate for SARS was about 10%.
So 1 in 10?
Mm-hmm. There were about 8,000 cases worldwide and 774 deaths. Okay. With the novel coronavirus, we know how many people have died.
Yes.
The trouble is we don't know how many people have been infected.
Right.
And so we're still in the process of defining how wide the pyramid is. We know what the top looks like, but in any disease there's a pyramid that starts from mild infections to more serious, those that require medical attention, hospitalization, and severe illness and death. So we now know a bit more about the top of the pyramid, and that's why I can't tell you today what the case fatality rate is.
Fair enough. Today, if someone got this coronavirus and had the illness and showed up at the hospital and was diagnosed, what would be the treatment that would be offered to them?
Well, first of all, you'd want to exclude any other treatable secondary infections or co-infection, and otherwise it's essentially supportive treatment, which might include oxygen, ventilation, etc. There are some experimental treatments that are being actually initially discovered with SARS, tested in vitro for SARS and MERS, and so potentially might be relevant for N coronavirus, but we don't yet know that they will really work in that setting. And interestingly, one of them is an HIV drug, which is fascinating to me because it's one particular drug, not the whole class of drugs. It's a drug called Kaletra or lopinavir, and it works in vitro. It stops replication of SARS and
in vitro meaning in test tubes?
In test tube models,
works,
and MERS, not yet tested with coronavirus. And there's some suggestion that it was a benefit in SARS, not yet demonstrated in MERS, and being evaluated currently for the new coronavirus. There are other novel antivirals. So all of these viruses, including HIV, are RNA viruses, and RNA viruses use particular enzymes to replicate. And in looking for new anti-Ebola drugs, one of those drugs also works in coronaviruses, a drug called remdesivir.
So it seems that all the research we've done previously in SARS and MERS and Ebola is actually helping taking the first steps against the coronavirus.
Yeah, in so many ways actually, because these things come, especially SARS came, caused an enormous amount of disruption on so many levels and then went away. And the worst thing we could do is just move on. You need to learn and respond. And actually I think something that changed the way we are prepared for new outbreaks was Ebola. So Ebola in 2014 was the worst outbreak, killed 11,000 people, and there was an increased awareness around the world that we are not ready, and it's very difficult to respond and develop new vaccines and drugs and test them in the middle of an epidemic.
Right.
And one of the spin-offs from Ebola was increased driver for investment in new vaccines and new antivirals. And it's very interesting, I think, what's happening in the vaccine world. An organisation was established for this very reason, and their goal was to be successful with funding from an organisation called CEPI, was that you had to have a vaccine developed within 16 weeks. And those
16 weeks of the start of a disease, so to say, you'll have a vaccine.
You'll have a
for it.
vaccine.
In what we know now, what do you see the trajectory of this virus being? So today it's about 25,000 people or thereabouts reported as cases, 500 deaths reported, most of them in China, most of them in the original province. How do you expect this will play out?
I don't think the disease has peaked yet. I think it will peak in the next several weeks. And I think that we are in for at least several months of seeing this.
Right.
Because unlike SARS, there's community spread, and it's much harder to control community spread.
Right. Even though they've locked down cities, I understand it's almost an unprecedented public health intervention.
It certainly is, and it'll be very interesting to see how effective it is. In the past, closing borders can delay the spread and delay the importation, but it doesn't totally prevent it, because it is very difficult to have tracked down every single traveler. So these measures delay and give you time to prepare.
Right.
But if there's true community spread, it's very difficult to keep something out.
And by community spread, you mean where humans give it to other humans outside of a healthcare setting?
That's right.
Which it is quite clear that
That's
with this
right.
virus.
With SARS and MERS, almost all the spread was with close contact and in healthcare settings. So once you figured out and could identify people, you could institute control measures to prevent spread.
Right.
You could institute measures within healthcare facilities as well as in close contacts. But if there's spread beyond that, or if very mild cases exist in large numbers,
Right.
and if they are sources of spread, then it becomes that much more challenging.
From a public health perspective, let's start with the first thing, masks. How effective are masks? I'm not talking in the hospital for doctors and nurses and for known patients, but how effective is this business of wearing masks going around daily life?
So there's not a lot of evidence to suggest that healthy people get any benefit from wearing a mask. They're not protected by wearing a mask if you're healthy and aren't infected. It makes more sense if somebody is already infected and has respiratory symptoms and they want to sort of contain their cough. For them to wear a mask might make more sense, but it doesn't actually make much sense for a perfectly healthy person to walk around wearing a mask with the idea that they're protecting themselves.
Most people on the street are wearing surgical masks, which are the
Right.
simple
In any case, yeah.
masks you can buy in any pharmacy and quite cheap, and they get very moist very quickly, and
Yes.
they don't protect you from viruses can get through that.
Right.
In the
So-
hospital setting, we do use masks. In fact, people are told that if you're symptomatic,
Yes.
call your doctor or call the hospital, and as soon as you arrive there, they'll put a mask on you.
In
to
fact, they're
prevent-
telling people if they have a mask to arrive with the mask
Yeah.
on,
So they have some role, but not used.
but that's when
in
they're
the
they're
public.
already unwell.
Right, but not as a public health measure that everyone goes around wearing masks. At the
Exactly.
moment, there is not much reason to do
Yes.
that.
In fact, good hand washing is much more important. And
Hand washing and
cough etiquette, cough and sneezing.
Coughing and sneezing etiquette.
Yeah, and when you are in a hospital, we do use a different type of mask to a
It's a
surgical
special-
mask, a
right.
special N95 mask, which is thicker and fits appropriately around your face. Yeah.
Do that. Now, of course, the hope would be that this would either naturally subside, and if it doesn't, that we can contain it effectively with perhaps a vaccine or treatments. Given what you have learned from all the other viruses, how likely do you think, and what time frame should one expect these things to take?
For development of
Yeah.
cures or vaccines. Well, I mentioned earlier about vaccines that most people think to develop a vaccine may take you 2 or 3 or 5 years, and I think given the significant investment that's been made literally over the last 5 years, we may see a vaccine in clinical trial, of course. You may develop it in 16 weeks and then it needs to be in clinical trial. To work out whether a vaccine works, you need ongoing transmission. So
Yes.
you'd have to trial it. So we may see a vaccine, best case scenario, could be under a year, then you've got to manufacture it and roll it out. It's interesting with influenza, there's been a lot of discussion about developing vaccines for a pandemic strain.
Yes.
And what's the minimum estimate still at 6 months, even when you know exactly what your vaccine is going to look like to
Right,
make
and then of
it.
course the
Well,
influenza
that has
cycle
to do
moves
with
on. Yeah.
right, but the influenza story has to do with the method by which our vaccines are
Yeah.
made. There are many new platforms that are being investigated for novel viruses like this. And so that time span can be shrunk quite significantly. But the issue is that we don't actually have vaccines that are licensed using those platforms.
Right.
So the safety data that will be needed will be much
Right.
greater.
Right.
And then we'll have to know whether they work.
Right.
And we have to know that they don't produce any harm.
Right.
So there are all these other
So it's a long
coronaviruses.
business. So
it
if
is a
they
long
start
business.
in 16 weeks, but to scale
Yes.
it up to make sure it's safe and then to
Exactly.
distribute
Safe
it
and
out
effective
will take
and-
time.
yes.
Well, from a treatment perspective, how long do you think before we actually have treatments for people who have this?
Effective treatments may be much quicker, I think, because we have a few lead candidates one drug is already licensed. Interferon is another drug that could potentially work. Remdesivir is not licensed, but we have a few lead candidates that are actually in clinical trials right now for the novel coronavirus. Many people will be screening drug libraries. That was the big breakthrough for sharing the virus from the Doherty, is that it allows people to grow the virus in laboratory models
And test their drugs on them.
and test an antiviral.
So
to test whether
isolating
it
the
works.
virus was a very important step in
Very
assisting those treatments.
Yeah.
Well, there was something you said, Kanta, that worried me. Is that once you said that it was only around 2003 or thereafter we started seeing these big transmissions from animals to humans. But now they seem to be happening more commonly, because this one is also tracked back to bats. So what changed in our relationship to animals?
So a lot has changed in our relationship to animals. We're taking away their habitats all around the world. And so we are seeing many more zoonotic infections. There's much more of a recognition of the importance of One Health, that we cannot achieve human public health without veterinary public health and without paying attention to the environment. So that is something that has come as a result of these outbreaks that have been all traced back to zoonotic infections, including Ebola, SARS, MERS, and so on. So I think that is something that we all must recognize and learn to deal with, because there is an enormous reservoir of pathogens in animal hosts, and if we take away their habitats and live in much closer proximity, that's a problem. With the coronaviruses in particular, all three of these have come from animal contact with people. And at least two of them, SARS and this one, appear to have been traced to wet markets in southern China. So one quick way to deal with things, which will, quick I say in a quick decision, but very difficult to implement, would be to close markets that trade in wild animals in proximity to people, especially live wild animals. But then again, that will take a long time to achieve, but clearly
and
that
may
is
be
a source.
to enforce.
But if you have 100,000 people infected, maybe it's time.
I mean, large urban centres and travel make a big difference
Yes,
too.
and then of course.
So the previously SARS in southern China, which we all know about, large urban centres, and then a big outbreak in Hong Kong.
And Hong Kong had the first bird flu, H5N1 infections, and they have live markets, they still do, but they've instituted a process where they close the markets once a month and clean them. And that has really
so different
controlled.
hygienic practices.
Yeah, so it is possible to clean up some of these markets, but whether we really should be having bats and civets and other animals all together in one market.
True. And so the Doherty Institute was right at the forefront, ready for this challenge. But what is the Doherty doing now, as this challenge will likely move, and particularly in the direction of One Health and interaction with animals?
Well, I think the whole conversation that we've had today, talking about SARS and MERS and H1N1 and coronavirus over just the last 17 years, tells us we will have new epidemics, so we must learn from each one of them. And I think there's an enormous amount to do for this one now. And so the Doherty is quite unique because of its breadth of expertise. And so we have some fantastic work happening in public health. Many of our staff are advising the government daily on policy. So when policies come out, like closing the borders to China, these are not made lightly, and many of our experts here
have contributed to that.
Have contributed to those discussions. We also are doing a lot with the benefit of having grown, well, we'll be doing a lot with the benefit of having grown the virus right here, which, as we talked about earlier, will drive, we hope, the development of finding novel antiviral drugs, novel diagnostics, and potentially an animal model. An animal model will be absolutely key to test these antivirals and vaccines. And in these very early days of a new infectious disease, there's so much to learn about the natural history, the immune response, the duration of shedding, the duration of being infectious. So really close experimental medicine, really close analysis of individuals who come in and come in with infection. Very, very valuable. And we've been the Doherty, many investigators across the Institute have been part of a network that was established after H1N1 in preparedness research for infectious diseases. It's called a PRIZE.
Right.
And part of that network is a number of clinical trials that have now been put into action. One of them is led by Irani Thevarajan, a infectious disease physician at Royal Melbourne, which is pre-approved ethics, which can sometimes take weeks, but it's already approved to collect specimens of anyone that comes into the Royal Melbourne with novel coronavirus, and hopefully we'll extend that across Victoria. And there's another fabulous network that is run out of the intensive care units across Australia that are also ready to go. Should any of our patients here with novel coronavirus get into intensive care, They're part of an international network that will not only characterise natural history of the infection, but are ready to go on novel interventions as well. Yeah.
Well, can I end by first thanking both of you for your expertise. As you can imagine, at this point in time, there are lots of questions in everyone's minds, and the newspapers can answer only so many of them. So thank you for taking your time. Also, thank you for institutes like the Doherty Institute, where you've prospectively thought of those things, where you brought together healthcare as well as scientists who are not only thinking about today's problems, but tomorrow's problems. Makes the world a bit safer. Thank you so much.
Thank you.
Thanks, both.